Hello class, here is Dr. Pixie. Welcome to the elementary system MDR3008. Development of the midgut. Before we discuss the development of GIT, it would be great that we begin with the fertilization. In week 1, a sperm fertilizes the ovum and gives rise to the zygote. The zygote then undergoes repeated cell division to form the blastula, which is a sphere of cells. In week 2, the blastula implanted into the uterine wall. Following that, in week 3, the blastula turns into gastrula through the process called gastrulation. Gastrulation then gives rise to 3 germ layers. The 3 germ layers are the blue color ectoderm, the red color mesoderm, and the yellow color endoderm. The ectoderm will give rise to the future central nervous system and the skin. The mesoderm is divided into 3 components. Here is the notochord in the median section. The mesoderm closest to the notochord is called paraxial mesoderm, which will give rise to muscle and somites. Then the intermediate mesoderm will give rise to kidney and the gonads. Further on the lateral side, the mesoderm is called the lateral plate of mesoderm. It divides into 2 parts. For that, we'll look into this embryo. The lateral part of the mesoderm is further divided into the parietal mesoderm and the visceral mesoderm. The parietal mesoderm is also known as the somatic mesoderm, which will then give rise to parietal peritoneum. On the other hand, the visceral mesoderm is also known as the splenic mesoderm, which will give rise to visceral peritoneum and the smooth muscles of the GIT. Further on, the yellow color endoderm will then give rise to the epithelial lining of the GIT. Now, what you can see here is the yolk sac, which is a source of nutrient for the developing embryo. During this time, the developing embryo is having the yolk sac connected to the midgut via the vitelline duct. In between week 3 and 4, the lateral folding of the embryo is happening. And eventually, the ectoderm. Here is the notes to describe the developing midgut, which will then form the U-shaped primitive intestinal loop. We are going to discuss this by using the diagram. Here is the diagram showing the 5-week-old developing embryo. Here is ventral, dorsal, cranial, and caudal. In the longitudinal section through the 5-week-old embryo, we can see that here is the developing gut tube. Here is the rapidly growing midgut, which is forming a U-shaped primitive intestinal loop that has 2 limbs. Here is the vitelline duct connecting the midgut into the yolk sac. So the limb above the vitelline duct is called the cranial limb. The limb below the vitelline duct is called the cranial limb. So this is the slide that's summarizing the derivatives of the midgut, and I divide it into the derivatives according to the cephalic or cranial limb and the caudal limb. It is important to remember that the caudal limb gives rise to part of the ileum, the distal part of the ileum, cecum appendix, ascending colon, and the proximal two-thirds or the right two-thirds of the transverse colon. During the development of the midgut, it is important to remember is two stages of gut rotation. The first stage of rotation will rotate 90 degrees counterclockwise around the axis of SMA. This is important to remember. Following that, the second stage of rotation will involve another 180 degrees of counterclockwise rotation, making a total of 270 degrees of rotation. And we are going to discuss all this by using the diagram. When we talk about the development of the midgut, it is always important to remember the physiological umbilical herniation, which means it occurs during the normal condition. During development, the midgut forms a U-shaped primitive or primary intestinal loop. This loop has two limbs, and using the vitelline duct, which is connecting to the yolk sac, as the landmark, the limb before the vitelline duct is called the cephalic limb or the cranial limb. The limb after or below the vitelline duct is known as the caudal limb. So during the development, the midgut communicates with the yolk sac, which is a source of the nutrients to the embryo, through a narrow stalk. This stalk is known as vitelline duct or vitelo-intestinal duct until week 10. And this umbilical herniation occurs is because the rapidly growing midgut is not able to fit into the developing abdomen with the limited capacity. And this is due to the abdomen is mainly occupied by the relatively massive liver and the two kidneys that are exit during this period of time. After the physiological herniation of the small intestine into the umbilical cord, by week 10, this part starts to re-enter into the abdominal wall. It begins with the right limb of the developing intestinal loop and followed by the left limb that re-enter into the abdominal cavity with the cecum as the last structure to return. In this diagram, we are going to describe the development and the rotation of the midgut. It is always important to remember that the midgut will undergo two stages of rotation. First stage, it rotates counterclockwise 90 degrees. Second stage, it further rotates for 180 degrees counterclockwise. This rotation occurs along the axis of the superior mesenteric artery SMA. This is showing the 5th week old of the embryo with the stomach, midgut, and hindgut. The midgut is communicating with the yolk sac via the yolk stalk or the vitelline duct. In this magnifying diagram, we can see that this is the developing primary intestinal loop with its vitelline duct connecting to the yolk sac. The limb that is above the vitelline duct is called the cephalic limb or cranial limb. The limb that is below the vitelline duct is known as the caudal limb. And here's the developing diverticulum called cecum. Here, this is the cross section that we can see that this is showing the SMA is the superior mesenteric artery that is the axis for the rotation. And here above it is the cranial limb and below is the caudal limb. So during the second month of development, and to be exact is around week 6 of development, the midgut is undergoing a rapid growth in which the loop now is undergoing a physiological herniation into the umbilical cord, and it herniates outside the abdominal cavity due to the limited space in the abdominal cavity. And please remember that this herniation is physiological, which means it occurs normally. And during this herniation, the midgut will undergo the first stage of rotation, that it will rotate counterclockwise for 90 degrees, making the cranial limb now become the right limb, the caudal limb become the left limb. And how do you remember the rotation is counterclockwise CC? So I always remember that the midgut gives rise to cecum C and also the C shape of the duodenum. So for that, I always remember that the midgut rotates counterclockwise CC around the long axis of the SMA. After the physiological herniation in second month, following that in the third month, or to be exact is the week 10, this intestinal loop starts to re-enter into the abdominal cavity. And during this re-entering, it further rotates for 180 degrees. This is another 90 degrees, following by another 90 degrees. So it will rotate counterclockwise 180 degrees. Here is showing the 10-week-old embryo that the small intestine will return first, which means the right limb will re-enter the abdominal region first. It passes posterior to the SMA to occupy the central region over here. It occupies the central region of the abdomen. Following that, as the large intestine starts to re-enter, which means is the left limb or the caudal limb, when it re-enters, it undergoes another 90 degrees of the rotation. And when the caudal limb returns, the last structure that returns to the abdominal region is the cecum. So this is the cecal swelling, in which the upper part expands and forms the cecum, and the lower part will elongate to form the appendix. So again, during the physiological herniation, the intestinal loop undergoes 90 degrees counterclockwise rotation. Then in the third month, when it re-enters into the abdomen, the re-entering will cause another 90 degrees of the rotation, following by when the large intestine starts to re-enter the abdominal wall, it rotates for another 90 degrees. So altogether is 90 plus 90 plus 90, 270 degrees of rotation. In this slide, we further discuss about the cecal diverticulum, which start to appear during the 6th week of the embryonic development. The diverticulum appears on the anti-mesenteric border of the caudal limb of the midgut. Now, the apex of the cecal diverticulum doesn't grow as rapid as the rest. Therefore, the appendix, which is growing here, is initially as a small diverticulum of the apex of the cecum. The appendix is then increases rapidly in length, so that at birth it is actually relatively a long tube arising from the distal end of the cecum. And after birth, the wall of the cecum starts to grow unequally, which then push the appendix to become on its medial side. At the beginning of the 10th week, when the limbs start to re-enter into the abdominal wall, the cecum is located temporarily higher up onto the right upper quadrant. Slowly during development, the cecum will then reaching to its final destination at the right iliac fossa.
This is a brief overview of the embryological rotation of the midgut. This is a brief overview of the embryological rotation of the midgut. Arterial blood to the gut tube is supplied by the abdominal aorta. The gut tube is composed of the foregut with blood supply from the celiac artery, the midgut with blood supply from the superior mesenteric artery, and the hindgut with blood supply from the inferior mesenteric artery. During the 5th week of development, the midgut starts to loop ventrally, herniating into the umbilical cord. This growth process creates much of the resulting length of the intestines. The cranial limb of this midgut extension will ultimately form the distal duodenum, jejunum, and part of the ileum. The caudal limb of this midgut extension will ultimately form the distal ileum, cecum, appendix, ascending colon, and 2/3 of the transverse colon. As the midgut lengthens, it also rotates 90 degrees counterclockwise around the axis of the combined superior mesenteric artery and umbilical cord. The cranial limb of the midgut, now on the right side of the umbilical cord, becomes highly convoluted throughout the next few weeks. The caudal limb of the midgut, now on the left side of the umbilical cord, grows a small cecal bud, ultimately becoming the cecum and appendix of the colon. During the 10th week of development, the midgut retracts back into the abdominal cavity and rotates an additional 180 degrees counterclockwise, for a total 270-degree rotation of the intestines. The duodenum, jejunum, and ileum are now inside the borders of the entire colon, which is composed of an ascending part, a transverse part, a descending part, and a sigmoid part.
Applied anatomy. Let's talk about the Meckel's diverticulum. This is a true diverticulum because it contains all layers of the small intestinal wall. It represents a persistent remnant of the omphalomesenteric duct, or the proximal part of the vitelo-intestinal duct. This duct connects the midgut to the yolk sac, which is present in embryo and usually obliterates during 6 weeks of intrauterine life or the 9th week of the gestation age. This diverticulum arises from the anti-mesenteric surface of the ileum. It is important to remember the rules of two applied for the Meckel's diverticulum. The incidence is about 2% of populations. The location of the diverticulum is usually 2 feet from proximal to the ileocecal junction. It measures usually about 2 inches in length. For the female and male ratio, remember we start with the longer one. The female ratio to male is 2:1. And the age of presentation is usually about 2 years old. There could be atopic tissue present in the diverticulum, and usually there are 2 types. It could be the gastric or pancreatic cells. These atopic cells are the pluripotent stem cells. Therefore, it can give rise to any type of cells. Gastric tissue is the most common presence in the diverticulum. Therefore, these cells can secrete acid and can lead to ulceration. Here, using these diagrams, I'm going to explain the vitelo-intestinal duct, VD, or also known as the omphalomesenteric duct, OMD. Here is showing the developing embryo. Over here is the primitive gut, and which is connecting to the yolk sac via the omphalomesenteric duct. Along the development of the embryo, the duct is getting narrower, but still connecting the primitive gut, in this case is the midgut, to the yolk sac. By the gestation age of 9th week, the omphalomesenteric duct or the vitelo-intestinal duct usually obliterates. If the duct doesn't close or obliterate, 2 conditions can occur. It could remain as the patent duct. Over here is showing the ileum, and here is the anterior abdominal wall with the umbilicus. If the patent duct persists, this is known as the vitelo-intestinal fistula. The second condition that could occur is the Meckel's diverticulum, in which only the proximal part of the VI duct persistent. Note that here there is no connection between the ileum and the anterior abdominal wall. So what are the clinical features of the Meckel's diverticulum? Usually there is asymptomatic, and if there is any clinical features that includes the painless rectal bleeding in the form of melena, which means the black tarry stools. Now let's discuss some applied anatomy related to the midgut. First is the congenital omphalocele, as shown here. Omphalocele is an abdominal wall defect at the base of the umbilical cord. The infant is born with a sac protruding through the defect, which contains the small intestine, liver, and large intestine. Omphalocele is always frequently associated with other birth defects, such as heart defects, imperforated anus, urinary problems, and genetic defects. Omphalocele is very similar to another condition called gastroschisis, except that the organs are enclosed in a sac. In the case of the gastroschisis, as shown here, it is a condition showing the extrusion of the abdominal viscera through an opening in the abdominal wall without involving the umbilical cord. And this happens is due to the incomplete closure of the lateral fold during the 4th week of embryonic development. Another applied anatomy is umbilical hernia. This is a protrusion of the intestine through an imperfectly closed umbilicus, and the hernia sac is covered by the skin and also the layer of the peritoneum. Another applied anatomy related to the midgut is sometimes the midgut could have developed without the rotation. For that, the caudal limb of the intestinal loop will return to the abdomen first, followed by the cranial limb. This is the opposite sequence of the returning of the intestinal loop. So now the small intestine will lie on the right side and the entire large intestine is on the left side. The twisting of the intestine, called volvulus, may obstruct the superior mesenteric artery and can cause infarction and gangrene. Development of the hindgut. The hindgut begins at the junction of the right 2/3 and the left 1/3 of the transverse colon. Then it terminates at the superior 2/3 of the anal canal at the level of the pectinic line. And all the derivatives from the hindgut is supplied by the inferior mesenteric artery. So when we discuss about the development of the hindgut, we are going to focus onto the cloaca and the division or partition of the cloaca. Here is showing the foregut, the midgut, and the hindgut. And the cloaca is the dilated terminal part of the hindgut in which it is endodermal lined. So it is now in contact with the surface ectoderm that we can see here. Here's the surface ectoderm, and this endodermal line cloaca is in touch with the surface ectoderm via the membrane called cloacal membrane. And there is no mesoderm in between them. So this is a bilayer structure. The cloaca is communicating with the hindgut, with the atlantoids ventrally, and the mesonephric ducts laterally. Again, it is important to remember the cloaca membrane is a bilayer structure formed by two germinal layers. First is the endoderm from the cloaca, then is the ectoderm from the proctoderm, which is the surface ectoderm. When we discuss about the development of the hindgut, it is important that we talk about the cloaca. Again, the cloaca is the terminal dilated part of the hindgut, and it is important to remember that the cloaca is further divided or partitioned into anterior part, ventral part, and dorsal part, the posterior part, by the urorectal septum. The urorectal septum is developed from the splenic mesoderm. It grows downward and then partitions the cloaca into two parts. Ventral part will then become urogenital sinus that will then further develop into different structures according to gender. In female, urogenital sinus will become bladder, urethra, and vestibule. Whereas in male, the urogenital sinus gives rise to bladder and prostatic urethra. Dorsal part of the cloaca is then become the anal-rectal canal, which is same for both sexes. The anal-rectal canal will further develop into rectum and the superior two-thirds of the anal canal at the level of the pectinic line. The urorectal septum continue growing downwards and towards to the cloaca membrane, in which at the week 7, it fuses with the cloaca membrane to form the primitive perineum, represented by a perineal body. Clinically, the perineal body is known as perineum. What is perineal body? The perineal body is the central tendon of the perineum. It is an irregular fibromuscular mass. In adult, it has a high content of the elastic connective tissue blended with some smooth muscles. It is located at the midline of the perineum. We're going to look at the perineal body in the next diagram. The function of the perineal body is it is an anchor point for many perineal muscles. It is critical for maintaining the integrity of the pelvic floor, especially in female. Further on, the urorectal septum partition or intersect the cloaca membrane into two parts. Ventrally, it becomes the urogenital membrane, and posteriorly, it becomes the anal membrane. The urorectal septum divides the cloaca sphincter into anterior ventral or the dorsal part as well. Anteriorly, the cloaca sphincter will become the superficial transverse perineal muscles, bulbous spongiosum and ischiocavernosus. Whereas dorsally, it becomes the structure related to the anus, that is the external anal sphincter. Using this series of diagrams showing the embryo at week 4, week 6, and week 7, we are going to explain about the partitioning of the cloaca, which is a very important part of the development of hindgut. In this diagram, the longitudinal section of the embryo, we can see that here is the dilated terminal part known as cloaca. It gives rise to two structures. It will give rise to GIT component and the urogenital component. The cloaca is in communication with a few structures. It is in communicating with the hindgut, allantois, and also laterally with the mesonephric ducts. Here comes the growing of the urorectal septum, which is a splenic mesoderm. The urorectal septum is having a downgrowth, growing downward, and slowly partition the cloaca, dividing it into the ventral part and the dorsal part. Then, coming to week 6 of the development, the urorectal septum is now slowly dividing the cloaca into the ventral primitive urogenital sinus and the anal-rectal canal dorsally. As we recall, the urogenital sinus will become different structures for different sexes, but for both will definitely give rise to bladder. For the anal-rectal canal, this part will become future rectum, and also the upper two-thirds of the anal canal up to the level of the pectinic line. Here, there's one structure that is not highlighted. It's called proctodeum, which is an inward folding on the surface of the ectoderm that develops into part of the anal canal. We were going to see that in the next diagram. From week 6, now we move on to the week 7 of the development, in which the urorectal septum has completed the partition of the cloaca, become ventral urogenital sinus, and dorsal the anal-rectal canal. Then, in week 7, the septum will fuse with the external ectoderm, forming the perineal body. And at the same time, the urorectal septum will then fuse with the cloaca membrane and partition it into now the cloaca membrane is being partitioned into the anterior membrane, the ventral membrane, becoming the urogenital membrane. And the dorsal part, this membrane will become the anal membrane. So by the end of the week 7, the embryo has separated the urinary system from the anorectal canal by the development of the urorectal septum fusing with the ectoderm, forming the perineal body. Here is another series of diagrams to show the partitioning of the cloaca by the urorectal septum. We are showing here the week 4 embryo, week 6 and week 7. What I want you to focus is the blue color region is the cloaca membrane. It is a bilayer membrane formed by 2 germ layers, inner endoderm and outer ectoderm. And this is the protoduum, in which there is the invagination of the surface ectoderm into the anal canal. Now in week 4, the urorectal septum is growing towards the cloaca membrane. In week 6, it is growing closer to the cloaca membrane and is slowly dividing it into the ventral urogenital sinus and the dorsal anorectal canal. And this is the growing phallus. At the week 7, the urorectal septum will fuse with the cloaca membrane to become the perineum or the perineal body. Then it also partitions the cloaca membrane into the urogenital membrane and the anal membrane. When these two membranes rupture, the ectoderm and the endoderm will become continuous with each other. In the urogenital triangle, the point of separation of this ectoderm and the endodermally derived structure is at the fossa navicularis in male urethra and at the hymen of the distal vagina in the female. The location of the cloaca membrane is approximately in the presence of the pectinic line in the distal one-third of the anal canal. It is always thought that the anal valves are the remnants of the cloaca membrane. Using this series of the diagram showing the embryo between week 4 to week 6, we are going to describe the subdivision of the cloaca into the ventral primitive urogenital sinus and the dorsal anorectal canal. This subdivision of the cloaca is achieved by the growing of the urorectal septum. The urorectal septum is derived from the splenic mesoderm. And with the 3 distinctive growing septae, is going to achieve the complete partitioning of the cloaca. So the 3 distinctive septae are the 1 superior fold and 2 lateral folds known as the rachis folds. So in this diagram is showing the dilated part of the hindgut, which is the cloaca. And right over here, this is the superior fold of the urorectal septum. And when it is migrating downward, it is going to meet with a pair of the lateral folds. These lateral folds will then grow sagittally to meet the descending urorectal septum. So 3 folds altogether, 1 fold from superior, 2 folds laterally, here 1, 2 laterally and 1 superiorly. When these folds interface with each other and the descending urogenital septum is completely dividing the cloaca into 2 parts, into the ventrally primitive urogenital sinus and also the dorsally anorectal canal. So as we recall, the urorectal septum has the upper portion and the lower portion. The upper portion is derived from the descending visceral mesoderm, and the caudal portion is derived from the lateral folds. Misalignment of the descending urorectal septum and the lateral folds will cause many abnormalities associated with the hindgut. Congenital abnormalities. The anorectal abnormalities can be subdivided into high and low depending on whether the bowel terminates above or below the puborectalis muscle. The first category is the urorectal septal defects. This will be involving either the both lateral folds or the failure of the superior and the lateral folds or the misalignment of the folds. When the lateral folds are failed with defects, this will cause the rectal urethra fistula. To be exact, in male, this will cause the rectal prostatic urethra fistula, and in female, it will lead to rectal vaginal fistula. When there is a defect between the superior septum and the two lateral folds, this will cause the rectal vesical fistula in males, and in females, this could disrupt the fusion of the paramesonephric ducts, producing pair of the bilateral vagina and the uteri that empty into the bladder. The urorectal septal folds could be misaligned. In the misaligned, it will cause the urorectal fistula. In males, a fistula is formed between the rectum and the prostatic urethra. And in females, the rectal vaginal fistula is formed. Here are the diagrams showing the urorectal septal defects in the male and the female. In the male, this is what we see is the urorectal fistula, and here is the rectal perineal fistula. In the female, this is where the part of the rectum is connecting to the vagina is known as the rectal vaginal fistula. And here is showing the imperforate anus. The second category is the low anorectal defects. This is the abnormalities involve the proctodeum and the anal pit that forms the distal one-third of the anal canal. That means it's below the pectinic line and the anus. The first type is the imperforate anus. This is the condition either the failure of the anal portion of the cloaca membrane rupture or it ruptures incompletely. The proctodeum fails to become continuous with the endoderm of the hindgut, which means the rectum is not properly connected to the body wall. The imperforate anus occurs 1 in 5,000 births, and it has a higher incidence in males. Anal agenesis is also known as the insufficient anus. The anal genesis could happen without fistula or with fistula. If the anal agenesis without fistula, the rectum will end blindly, with the location of the anal pit sometimes marked by a small dimple or pigmented spot. For the anal agenesis that occurs with fistula, the fistula will grow into the urethra or vulva, and this is due to the failure of the urorectal septum to divide the cloaca properly. For the high anorectal defects, that includes the anal rectal agenesis that involves the rectum, the rectal atrachia, and the persistent cloaca. The diagram A and D are showing the normal partition of the hindgut in females and male, and here are showing the congenital abnormalities involve the hindgut in the female and the male. Here, B and E are both showing the anal opening missing or closed off. And below here is showing a fistula which is occurring at the higher level. So here is the rectal vaginal fistula, and here is the high rectum connects into the bladder known as the rectal vesical fistula. Here is a series of diagrams to show the congenital abnormalities of the hindgut in male and female at the level of low or high in reference to the level of the puborectalis sling. Thank you.